Welcome to Chapter 4 of Concepts of Biology. We're going to be talking about energy production, how cells make energy, how they store energy, all of that stuff. Energy. You need it for everything. Living things need energy to do anything. In this image, what you're seeing here is a hummingbird getting their little sugar and things from plants. Hummingbirds use an incredible amount of energy for how small they are because of how quickly their wings have to move. So hummingbirds are almost constantly eating just so that they can maintain their energy stores to do what they need to do, such as fly and eat more sugar. Energy metabolism. So today we are going to talk and explain what metabolic pathways are. We're going to talk about the first and second laws of thermodynamics. We're going to talk about the differences between kinetic and potential energy. We're going to describe endergonic and exergonic reactions. And then we're going to talk about enzymes briefly and how they work as catalysts for different things that need to happen. So bioenergetics. What is bioenergetics? Bioenergetics is the concept of energy flow through living systems. Essentially, that just means how does energy flow through living things? Through plants, animals, humans, whatever. I mean, humans aren't animals, but that's a different conversation. That's bioenergetics. We will find that most things on Earth that need and use energy actually get their energy from the sun. Plants grossly outweigh the number of animals that are on this planet, so the majority of living things will get their energy from the sun. Those plants will grow and die. Some animals will eat those plants, some other larger animals will eat those animals, and it becomes a whole cycle. We call this the life cycle. And we'll talk about different levels of producers and consumers a little bit later on in the course. But for now, what you need to know is that ultimately, most life forms will get their energy from the sun. Herbivores will eat those plants, carnivores will eat those herbivores, and then eventually decomposers like fungus, bacteria, worms will eat us all and recontribute to the cycle. Okay. Metabolism. We hear this word metabolism often, that people have a slow metabolism, they have a fast metabolism. But what exactly is metabolism? Metabolism is all of the chemical reactions in a cell. When we talk about metabolism, we talk about either anabolic metabolism or catabolic metabolism. Anabolic metabolism is the process of adding things together to store energy. And catabolism is breaking things apart to release energy. The way I like to remember these things, anabolic kind of sounds like adding, add, keep those two together. And the way I think about catabolic is that cats break things. If you have cats, then you know they break things all the time just for fun because they're jerks. But I digress, and that's how I like to remember them. Catabolic is breaking them, anabolic is adding them back together. Like I said, this is kind of that anabolic and catabolic processes. So anabolics will add things together to store energy, require energy. Catabolic breaks them down, and you get energy as a product. Thermodynamics. Thermodynamics is the study of energy and energy transfer in physical matter. So anything that is made up of atoms, because remember, atoms matter, right? What is energy? Energy is scientifically, we label energy as the ability to do work. Work capital. Work being literally anything. Eating, moving, thinking, sleeping, dreaming. All of those things are work, classified as work. Okay? There are 3 different types of energy that we talk about. We talk about electrical energy, we talk about light energy, and we talk about heat as energy. Okay? There's a formula that equates work and energy, but that's a physics thing, and we're not going to think about that right now. Okay? So those are the types of energy that we talk about. There are a few laws of thermodynamics that we need to get kind of clear in our head. The first law of thermodynamics states, and this is the quote of it all, the total amount of energy in the universe cannot be, or is constant and conserved. Constant and conserved. What does that mean? That is the all energy can neither be created nor destroyed law. According to the first law of thermodynamics, energy can be transferred from place to place, it can be transformed, but it cannot be created or destroyed. So you can transform it, you can move it, you can transfer it, but you cannot make it or destroy it. These are just some examples of energy being transferred or transformed, right? When you eat a bunch of ice cream, you eat a bunch of sugar, all that stuff, that all of that chemical energy in that food gets transformed into kinetic energy, which will then get used by these, in this example, by these kids, by riding their bikes. They're using energy to power the pedals so that they can move the bikes. All of that is work that is done by that energy. Okay. And that energy will then again be transformed into power of the bike moving and heat from these kids sweating. So even then, that energy is still being transformed in different ways, transformed and then used. And then same thing with the light. The light energy from the sun transforms into chemical energy by these plants via the process of photosynthesis, which we will also talk about. Potential and kinetic energy are exactly what they sound like. Potential energy is the possibility of energy production. So like still water doesn't move very much, but it has a lot of potential energy because it could be moving very quickly, right? When you're holding up a book up high, it has potential energy when it's before it drops to the ground. Kinetic energy is the energy that you use when it is actually moving. So kinetic energy is your actually moving energy. And potential energy is your possible movement energy. Okay. Like batteries, for example, batteries store a lot of potential energy. And not until you put those batteries in the remote and you're using the remote to do things will it be start transforming into kinetic energy. Okay. Um, Chemical reactions, some processes are endergonic, meaning that they will take energy to complete. And other reactions will be exergonic, meaning that they release energy. Okay. So endergonic, exergonic, very similar to kind of anabolic, catabolic, just different types of words using that essentially say something very similar. Next, we're going to talk about enzymes. Enzymes are proteins. Let's get that clear right off the bat. Okay? They are made of amino acids. Therefore, they are proteins. Enzymes are catalysts for reactions, meaning they help start reactions. Okay? Without enzymes, the things that our body needs to do would still happen, but they would take forever. It would take so long for anything to happen. Any of the ATP production, any of the glycolysis that we're going to talk about pretty soon here, all of that would take so long without enzymes. So how do those enzymes speed up that process? Enzymes speed up the process by lowering what we call the activation energy. If you've ever been like sitting on the couch and you know that you have something to do and it's that your deadline is getting closer and closer and closer, but you just don't want to do it, at some point you will reach the threshold of your activation energy where either the panic sets in and you have to start doing the thing, or the deadline comes and you have to start doing the thing. Either way, something is going to force you to get up and start doing the thing that you need to do. That's how you can kind of think about activation energy. So enzymes use are part of their function is to lower the threshold of activation energy. So going back to our panic example, um, if you had an enzyme that helped to lower the stuck in the couch feeling, then that would be considered your enzyme, right? So lowering the activation energy doesn't change how much energy is produced by the reaction. It just produces how much energy it takes to start doing the reaction. Enzymes use very specific active sites. The active site will be kind of like in this example, it's right in here. So the active site will bind to a very specific substrate. The enzyme will then slightly change the substrate and then throw out products. Sometimes the product will help to stop the original process, but that's a different kind of enzyme regulation. But essentially, that's how enzymes work. Specific substrate, specific active site, they create that enzyme substrate complex, and then the products pop out. Okay? It's kind of a lock and key mechanism. When we talk about regulating enzymes, there's a couple of different ways that they can be regulated. So first, allosteric inhibition. This is a process where the enzyme activity is either slowed down or deactivated, and it's usually done so by a inhibitor molecule, a molecule that comes in and changes the shape of the active site so that it no longer fits the specific substrate. And in that case, then the enzyme substrate complex will not be created and the enzyme does not do its function. So this is a form of competitive inhibition. There's also non-competitive inhibition. And non-competitive inhibition is essentially just where an enzyme will bind with something at a non-competitive site and it just changes the shape of the enzyme because that's what it's supposed to do. So there's some molecules that have active sites for different molecules and can change based on those. Yeah, have you ever thought about how pharmaceutical drugs are made? They're made using, they're made based off of general metabolic pathways. Metabolic pathway refers to pretty much anything that happens in a cell to make or break energy. So even making medications is based on some of these metabolic pathways that happen in our cells. This kind of just showing how several enzymes or several substrates can be catalyzed by multiple enzymes. And that's kind of what I was talking about, how the end product can sometimes inhibit the process overall. This is negative feedback inhibition. Feedback. Okay, so let's talk glycolysis. Glycolysis is one of 3 big processes that our cells do to make energy. So we're going to talk about how ATP is used by the cell as an energy source. And then we're also going to talk about the molecules produced by the breakdown of glucose by the process glycolysis. If we break up this word, glyco means glucose, lysis means to break. So just the process of breaking down glucose. Okay. Glycolysis is a major exergonic reaction. Exergonic means that it will release energy. Okay. That energy is formed by the breakdown of glucose to release that energy as ATP. And remember, ATP is our body's energy currency. Okay. Most glycolysis will occur in the cytoplasm. Most prokaryotes, but all eukaryotes. So glycolysis, if this is our whole cell, and this is our mitochondria. This is the biggest mitochondria in the world. Glycolysis happens out here. Okay. With glycolysis, we start with a single glucose molecule. Oops. And by the end of glycolysis, we have 2 pyruvates, 4 ATP, and 2 NAD+. We also put in 2 ATP to start the process. Okay. This is ATP. It is an adenine, which is one of our nucleic acids, with its ribose base. That's included in that nucleic acid, and 3 phosphate groups. We talked about functional groups a little bit in lab, but ATP is 2-ring adenine, that's this, 1 ring, 2 rings, a 5-carbon ribose, that's this guy right here, and then the 3 phosphate groups, A, T, tri, P, phosphate. Okay. Essentially, all you need to know here is that we start with glucose. A bunch of enzymes come in, make some changes to the different glucose molecules. It gets broken down into 2 molecules of 3-carbon pyruvate. And then that pyruvate will either go through the citric acid cycle or, if there is no oxygen present, it will go through fermentation, which also makes ATP, just not nearly as much. Speaking of the citric acid cycle, what is it? The citric acid cycle is the next part of energy formation, the citric acid cycle and oxidative phosphorylation. Okay, so the citric acid cycle is a process that will eventually turn that pyruvate into another molecule that will go into the oxidative phosphorylation cycle. It essentially creates a bunch of electron donors that will then go to the oxidative phosphorylation or electron transport chain. So we will describe the location of the citric acid cycle and the oxidative phosphorylation in the cell. We will talk about the outcomes of the citric acid cycle. I'm changing this oxidative phosphorylation because it's too many syllables to ETC, electron transport chain, in terms of the products of each. We're going to describe the relationship between glycolysis, citric acid cycle, and oxidative phosphorylation in terms of their inputs and outputs, what they make, and whatnot. Okay? So, first kind of things that start to happen when we leave glycolysis and start to enter the citric acid cycle. That pyruvate will be transported to the mitochondria because remember, glycolysis happens in the cytoplasm. So the pyruvate gets moved over into the mitochondria, and then cellular respiration, oxidative phosphorylation, electron transport, all the same thing will happen in the mitochondria. Aerobic respiration happens if oxygen is present. This is the more normal way of producing energy, at least for humans. We breathe oxygen. That oxygen helps us with that energy production. Okay. Most of the ATP is generated in that last kind of steps of oxidative phosphorylation, the aerobic catabolism of that glucose. So the oxygenated breakdown of glucose is what makes the most ATP. And the electron transport chain is the very last kind of component of that aerobic respiration, which is why we need oxygen to breathe. The end of the electron transport chain has ATP synthase, an enzyme, turn ATP or ADP into ATP. So it adds a third phosphate, and then we have our ATP. Just a little note, when you see this root or this suffix ACE, A-S-E, most of the time it's going to be an enzyme. Okay. And that generates about 90% of the ATP made during aerobic glucose metabolism. Okay. So oxygen plus sugar equals high ATP production. Okay. So a little overview of the cycle. Before it fully gets into the citric acid cycle, that pyruvate is formed into acetyl-CoA. It loses a carbon molecule, and that carbon molecule will then turn into CO2. It will get joined with some other oxygen molecules and form CO2. So CO2 is one of the products of the citric acid cycle. Total, you're going to get, or per pyruvate, you will get 3 NADH, which is an electron transport molecule, 1 FADH2, again, another electron transport molecule, 2 CO2 molecules, and 1 ATP per citric acid cycle. I'm sorry if you guys hear my cat screaming. And remember, each pyruvate or each glucose molecule gets broken into 2 pyruvates. So all of this gets multiplied by 2. And that's your total amount of products for the citric acid cycle. The electron transport chain is essentially just a bunch of oxidation reduction reactions. There's some enzymes in here. Again, unless you are planning on taking a biochem course or something like that, you don't need to know all of the names of these enzymes. The only one that you really need to know is the ATP synthase complex. This ATP synthase complex uses a proton gradient to make ATP. It's kind of like a crank that pumps out ATP. It uses that energy from those hydrogens to pump out ATPs. Almost like I'm realizing now that you can't see me, but it's like one of those old school window cranks that you used to have in your car. Okay. So this is an overall view of the whole process. You get your glucose, this is all in the cytoplasm. You start glycolysis, you make the things that you need to make. That pyruvate turns into acetyl-CoA, starts the citric acid cycle, and will then go into the electron transport chain. I know this says Krebs cycle. Krebs cycle is the same thing as the citric acid cycle. Okay, oxidative phosphorylation is the same thing as the electron transport chain. Science just likes to make many names for the same things. I don't know why. Fermentation. Fermentation is just anaerobic respiration, so without oxygen. Because sometimes your cells need oxygen, or your cells need energy, and you are not breathing as much as you should be because you are running up and down a hill, or you are underwater, or whatever it is. Okay? So we're going to talk about the differences between aerobic and anaerobic respiration, and then the types of fermentation that occur in animal cells. Okay. So fermentation usually only happens in animal cells if oxygen is not present. How do the cells get their ATP? This fermentation process. This fermentation process, it's not as efficient and effective as our electron transport chain. It doesn't make as much ATP. And it creates a byproduct called lactic acid. And that lactic acid can kind of collect in your muscles, and that's what kind of starts making that soreness happen. Also the use of the muscles themselves, but all of that is because of the lactic acid production because of the lack of oxygen. Okay, so lactic acid fermentation, pretty common in muscles that have been exhausted by use. So you still do that kind of glycolysis, but instead of those pyruvate going to the citric acid cycle, they go through the fermentation process where they make 2-lactate, and they still end up making a couple of NADH or NAD plus, which is that electron molecule, which is honestly the real star of the show here. Okay. So in non-oxygenated forms, that pyruvate will turn into CO2 and acetaldehyde, and that acetaldehyde will form into an ethanol. The ethanol in this case is lactate, which is an alcohol. Okay, this is a similar process that like plants go through to make things like wine, beer. It's just not with these specific molecules. But we do fermentation all the time. Okay. Some things function better without oxygen present, and so we call that anaerobic cellular respiration. Some bacteria, some cells do better in non-oxygenated waters, air, all of that stuff. Okay. So next thing we're going to talk about are ways that carbohydrate metabolic pathways, glycolysis, citric acid cycle interrelate with protein and lipid metabolic pathways. And then we're going to explain why metabolic pathways are not necessarily considered closed systems. Okay. Essentially, all this is saying the connection to other metabolic pathways is that all of these things are connected. Other sugars can play a role in creating those electron donors. Things like fructose, sucrose, they get broken down into smaller sugars. Everything will then get broken down into glucose so it can be used by glycolysis. Anything that you eat, drink, anything like that will eventually get broken down into its smallest form, which is that glucose monomer. Proteins, there's lots of proteins involved in the process of glycolysis, citric acid cycle, all of that stuff. Every molecule change from one to another is done by an enzyme, and those enzymes force this process to move much more quickly than it already would. And then as far as lipids are necessary to, keep our cell membrane flexible. Cholesterol is one of those molecules that helps with that. And acetyl-CoA is one of the more important lipid-based molecules that we kind of talk about in this process.